Welcome to the deep dive. Today, we're digging into one of your body's most ingenious survival tricks,
Mm,
how it keeps a steady supply of glucose flowing, even when you haven't eaten anything for hours. Think about it. Your brain, your red blood cells, they need glucose constantly. But what happens when the sugar runs out?
exactly. And for this deep dive, we're focusing squarely on gluconeogenesis and the maintenance of blood glucose levels.
Mm.
Our plan is really to walk you through this key chapter from Marks Basic Medical Biochemistry step by step. We want you to grasp the intricate pathways, the regulation, everything that ensures your body has this critical fuel. And we'll definitely look at what happens clinically when things go wrong. You should walk away knowing not just what gluconeogenesis is, but really appreciating why it's so vital for survival.
Okay, let's get into it. Our source material here gives us a really fantastic view of the mechanics and, well, the sheer importance of this, mainly happening in the liver, right?
Primarily, yes. The liver does the heavy lifting.
So let's start big picture. What's the fundamental problem gluconeogenesis actually solves? Why do we even need it?
Essentially, it's your body's emergency backup generator for glucose. Simple as that. Imagine you're fasting for a while, or maybe after some really prolonged exercise, your glucose from food just runs out. And some tissues, critically the brain and red blood cells, are completely dependent on glucose. They can't really use much else. So gluconeogenesis, happening mostly in the liver and a bit in kidney cortex too, is the pathway for making new glucose from things that aren't carbohydrates. It's a lifeline, really.
Okay, making new glucose from non-carbs. What are these non-carb compounds then? Where do they come from, if not from food sugars?
Yeah, good question. The main players in humans are lactate, glycerol, and certain amino acids, particularly alanine. These aren't sugars themselves, no, but the body has clever ways to, take their carbon skeletons and rebuild them into glucose. We can dive into where each comes from in a bit more detail later.
Okay, so the sources describe this kind of elegant balance of glucose levels. After you eat, glucose goes up. Some gets stored as liver glycogen. Easy enough. But what happens when we start fasting? Does the body just flip the switch to making new glucose instantly?
Not immediately, no. It's more gradual. Within maybe 2 to 3 hours after eating, the liver starts breaking down that stored glycogen. That's glycogenolysis. That releases glucose quickly into the blood. It's your sort of immediate reserve. But as you keep fasting, those glycogen stores start to dwindle. They're significantly depleted after, say, 30 hours or so. And that's when gluconeogenesis really ramps up and takes over. Eventually, it becomes the main and then the only source of blood glucose. It has to keep those vital tissues, especially the brain, fueled. It's quite a remarkable metabolic shift. And it's also very active during sustained exercise, by the way.
A metabolic baton pass, you could say. Now, here's where it gets really fascinating for me, the hormones. How do insulin and glucagon manage all this? How do they direct the traffic?
Ah, yes, the hormonal control is absolutely key. You can't understand this without it. Think of it this way. Insulin, which goes up after a meal, is the storage signal. Tells cells, take up glucose, store it as glycogen, maybe make fat. Glucagon, which rises during fasting, does the opposite. It's the release and produce signal. It stimulates glycogen breakdown, and critically, it stimulates gluconeogenesis, turning that lactate, those amino acids, that glycerol, into fresh glucose. So yeah, this hormonal tug-of-war really dictates whether glucose gets stored or made on demand.
Okay, so gluconeogenesis makes glucose, glycolysis breaks it down. It sounds a bit like it's just glycolysis running in reverse. Is it that simple? Just flip the enzymes?
That's a really common thought, but no, it's not quite that simple. While yes, many steps are just the reverse of glycolysis and use the exact same enzymes, there are three really crucial steps that are different. They're essentially irreversible in glycolysis. So gluconeogenesis has to use different enzymes to get around these roadblocks. These are the bypass steps. And this design is brilliant because it prevents a futile cycle. You don't want to be burning energy making glucose just to immediately break it down again.
Right, that would be incredibly wasteful.
Exactly. So imagine that metabolic road analogy again. Most of the highway works in both directions. But there are three one-way streets going down in glycolysis. Gluconeogenesis has to build these special bypass ramps using unique enzymes to go back up. It ensures traffic flows the right way when you need to make glucose.
That's a great way to picture it. Okay, let's talk about these bypasses then. What's the first major detour needed to make new glucose?
All right, the first one, and it's arguably the most complex, gets you from pyruvate back up to phosphenyl pyruvate, or PPP. In glycolysis, going down, that's just one step using pyruvate kinase. But going back up in gluconeogenesis, it's a two-step process involving two compartments. First, an enzyme called pyruvate carboxylase. This one lives inside the mitochondria. It converts pyruvate to oxaloacetate, OAA. It adds a carbon dioxide molecule, that's the carboxylase part, and it needs biotin, vitamin B7, as a helper.
Okay, step one in the mitochondria needs biotin. Then what?
Then step 2, an enzyme called phosphoenolpyruvate carboxykinase, or PEPCK. This one converts the OAA into PP. This step needs energy, but it uses GTP, guanosine triphosphate, not ATP, which is interesting. It uses GTP to provide the energy in the phosphate group for PP.
2 steps, starting inside the mitochondria. So how does that OAA, the product of the first step, get out? Doesn't gluconeogenesis mostly happen in the cytosol?
Exactly. That's a really sharp point. OAA itself can't easily cross the mitochondrial membrane, so it gets cleverly shuttled out. It's converted into something that can cross, like malate or aspartate. These molecules exit the mitochondria, and then once they're in the cytosol, they get converted back into OAA. And this shuttle, especially the malate route, is actually really important because it also carries reducing power in the form of NADH out into the cytosol. And you need that NADH for a later step in gluconeogenesis. It's elegant. Solves 2 problems at once.
Wow, okay. A shuttle system that also provides needed coenzymes. Very efficient. So what's the second bypass, moving further up the chain toward glucose?
The second bypass deals with fructose phosphates. It converts fructose 1,4,6-bisphosphate back to fructose 6-phosphate. In glycolysis, the enzyme PEPCK adds a phosphate using ATP. Gluconeogenesis uses a different enzyme, fructose 1,4,6-bisphosphatase. And instead of using or making ATP, this enzyme just performs a hydrolysis. It uses water to clip off that phosphate group, releasing it as inorganic phosphate. Simple removal.
Just clips it off. Got it. And the final bypass, the one that actually gives us free glucose that can leave the liver cell and, go feed the brain.
Right, the final hurdle. This is converting glucose 6-phosphate into plain old glucose. Here, the enzyme is glucose 6-phosphatase. Like the last bypass, it just hydrolyzes, clips off that phosphate group. And this enzyme is special. It's located in the membrane of the endoplasmic reticulum, sort of a separate compartment within the cell. Think of it like the final export gate. Once that phosphate is off, the free glucose can be transported out of the liver cell and into the bloodstream. This enzyme is also used when releasing glucose from glycogen stores, by the way.
Okay, so we've got the pathway, the key bypasses. But let's go back to the starting materials. You mentioned lactate, glycerol, amino acids. Can we trace their journey a bit more? How do they actually feed into this whole process?
Absolutely. Understanding the precursors is just as vital. Let's break them down. First
Sure.
up, lactate. Where does it come from? Mostly from anaerobic glycolysis. Think intensely exercising muscle that can't get enough oxygen, or red blood cells which have no mitochondria at all. This lactate travels to the liver, where it's converted back to pyruvate by lactate dehydrogenase. And boom, that pyruvate can enter gluconeogenesis right at the start. This whole loop, glucose from liver to muscle, lactate from muscle back to liver, that's the famous Cori cycle. It's metabolic recycling.
So cycling lactate. Okay, what about amino acids?
Amino acids. These mainly come from breaking down muscle protein, especially during fasting or starvation when the body is desperate for fuel. Alanine is a really big one. It's easily converted to pyruvate in the liver by an enzyme called alanine aminotransferase. Other amino acids can be converted into intermediates of the TCA cycle, the Krebs cycle, and those intermediates, like oxaloacetate itself, can then be drawn off to make glucose. There's also a cycle here similar to the Cori cycle called the glucose-alanine cycle. Muscle sends alanine to the liver, liver converts it to glucose, sends glucose back, another recycling loop.
Makes sense. And the last one, glycerol.
Right, glycerol. This comes from breaking down stirred fats, the triacylglycerols in your adipose tissue. When fat is mobilized for energy, glycerol is released as a byproduct. Glycerol travels to the liver and gets converted into dihydroxyacetone phosphate, or DHAP. Now, DHAP is actually an intermediate in glycolysis and gluconeogenesis, so glycerol kind of jumps into the pathway midway, bypassing those initial pyruvate steps.
So it's got a shortcut.
Pretty much, yeah. But what's really important to stress here is what doesn't work for making glucose, at least not in a net sense in humans. That's acetyl-CoA. You get tons of acetyl-CoA from breaking down fatty acids. But those carbons, when they enter the TCA cycle, are eventually lost as CO2. So you can't take the carbons from fat breakdown, run them through the cycle, and get a net gain of glucose at the other end. The math just doesn't work.
So you can't just live on fat and make all the glucose you need.
Not from the fatty acid part, no. Only that small glycerol backbone from the fat molecule could be used. That's a really key point in metabolism. You rely on protein breakdown and things like lactate when carbs are gone.
Wow. Okay, so it's this incredibly complex system, pulling materials from different places, using these special bypasses. How does the body control it all? How does it decide, okay, now we need gluconeogenesis, or stop, we have enough glucose? It sounds like it needs really tight regulation.
It absolutely does. It's exquisitely regulated. And the control happens at multiple levels. First, there's just substrate availability. Makes sense, right? If the building blocks aren't there, you can't build it. So when insulin is low and glucagon is high, or maybe stress hormones like cortisol and epinephrine are up, that signals your fat cells to release glycerol and your muscles to release amino acids. More raw materials become available to the liver.
So being stressed or skipping meals automatically starts providing the liver with the stuff it needs to make glucose.
Exactly. The hormonal signal mobilizes the substrates. Then the enzymes themselves are regulated. We talked about glucagon being high during fasting. Well, glucagon sets off a signaling cascade inside the liver cells. This leads to several key changes. For instance, pyruvate dehydrogenase, the enzyme that usually sends pyruvate towards energy production via acetyl-CoA, gets switched off. This forces pyruvate towards gluconeogenesis instead.
Shuts one door, opens another.
Precisely. And at the same time, pyruvate carboxylase, that first bypass enzyme, gets switched on. It's activated by high levels of acetyl-CoA, which build up when you're burning a lot of fat. It's like the cell saying, okay, plenty of fat energy here. Let's use this incoming pyruvate to make glucose for the brain.
That's clever signaling.
It is, and it goes further. The actual amount of some key enzymes changes. Glucagon and cortisol can actually tell the cell's nucleus to make more PEPCK enzyme. That's called enzyme induction. You literally get more of the enzyme protein. And crucially, to prevent that futile cycle, glucagon signaling also leads to the inactivation of pyruvate kinase, the opposing glycolytic enzyme, keeps the flow going upwards towards glucose.
So it's regulating the activity and the amount of the enzymes, a multi-level control system.
Exactly. Similar things happen further up the pathway too, regulating fructose, 1,4,6-bisphosphatase, and glucose-6-phosphatase, ensuring they're active while their glycolytic counterparts are shut down during fasting. It's all about ensuring a net flow towards glucose production without wasting energy. A metabolic masterpiece, really.
It really is. And understanding these mechanisms makes the clinical cases in the source material so much clearer. They really show what happens when this delicate balance is broken. Let's talk about LM, the patient with alcoholism who came into the ER with severe hypoglycemia.
Ah, yes. Al's case is a textbook and tragic example of gluconeogenesis failure. He hadn't eaten for days, so his glycogen stores were totally gone. He was relying entirely on gluconeogenesis. But heavy ethanol metabolism drastically shifts the balance of NADH and NAD+ in the liver. You end up with way too much NADH. Think of NAD+ as being needed for key steps in gluconeogenesis, like converting lactate to pyruvate or glycerol phosphate to DHAP. If all your NAD+ is tied up as NADH because of alcohol breakdown,
Those steps just can't happen.
they just stop. The high NADH essentially blocks the entry of major precursors into the pathway. His liver couldn't make glucose. That's why his blood sugar plummeted to 28 mg/dL, causing those severe neurological symptoms: confusion, combativeness, seizures. His brain was literally starving.
Devastating. Okay, what about Emma W? She got hyperglycemia, high blood sugar after getting high-dose steroids for asthma. How does that connect?
Emma's case shows the opposite effect: gluconeogenesis in overdrive. Glucocorticoids, the type of steroids she received, like prednisone, are known powerful inducers of gluconeogenesis. They actually signal the liver cells to produce more PEPCK enzyme, one of those key bypass enzymes we talked about. So her liver just started pumping out way more glucose than her body needed, leading to that high blood sugar, 275 mg/dL, and the classic symptoms of hyperglycemia like excessive thirst and urination. It mimics diabetes in a way.
So steroids can directly crank up glucose production. And then there was Dianne A, who went into a coma after taking insulin but skipping her meal.
Diane's situation's different again. That's a hypoglycemic coma, but caused by too much insulin activity. Insulin's job is to lower blood glucose. It tells cells to take glucose up, and importantly, it strongly inhibits both glycogen breakdown and gluconeogenesis in the liver. So she took her insulin, expecting to eat and cover it with glucose, but then she didn't eat. The insulin hit, shut down her liver's glucose output, and pulled glucose out of her blood rapidly. Her blood sugar crashed.
A very rapid, dangerous drop.
Extremely rapid. And it's important clinically to distinguish that from something like diabetic ketoacidosis, DKA. In DKA, you usually have very high blood sugar, ketones, dehydration. Diane's symptoms like flushed, moist skin point towards acute hypoglycemia from excess insulin.
It really highlights how critical the balance is. The source material also touched on the long-term issues with high glucose and diabetes.
Yes, absolutely. While those acute highs and lows are dangerous, chronic hyperglycemia, like in poorly controlled diabetes, causes damage over time. The high glucose leads to something called non-enzymatic glycation. Sugar molecules basically start sticking on to proteins throughout the body, especially in blood vessels. This messes up the protein's structure and function. Over years, this contributes to serious microvascular problems, damage to eyes, retinopathy, kidneys, nephropathy, nerves, neuropathy. And it also accelerates macrovascular disease like heart attacks and strokes. Landmark studies like the DCCT and UKPDS prove that tightly controlling blood glucose significantly reduces the risk of these long-term complications. It underscores why managing blood sugar is so vital.
Okay, let's try and synthesize all this. If we were to boil down the key takeaways from this deep dive into gluconeogenesis, what would they be?
Well, I think first and foremost, gluconeogenesis is the body's essential pathway for making new glucose from non-carb sources, primarily in the liver. It's absolutely critical during fasting or prolonged exercise.
And crucially, it's not just glycolysis in reverse. You have those three key bypass steps using different enzymes, which allow for regulation and prevent waste.
Exactly. Those bypasses are key. And the regulation is incredibly sophisticated, involving hormones like insulin and glucagon, the availability of substrates like lactate and amino acids, and direct control over those key enzymes, all working together to avoid those futile cycles.
And we saw very clearly how disruptions from alcohol, from medications like steroids, from insulin imbalance can have really serious immediate clinical effects. It just highlights how vital this whole glucose balancing act really is.
Couldn't have said it better myself. Understanding gluconeogenesis really gives you an appreciation for the body's incredible adaptability and survival mechanisms. It's this constant, elegant process of recycling carbon to keep essential functions running no matter what.
It really is amazing. What stands out most to you from revisiting this? For me, it's just the sheer precision of the control, how the body fine-tunes glucose levels under such varying conditions. Makes you appreciate breakfast a bit more, doesn't it?
It certainly does. And maybe next time you find yourself feeling surprisingly energetic hours after your last meal, you'll have a little more appreciation for those intricate pathways, those bypasses and regulators, working tirelessly behind the scenes in your liver to keep your brain happy.
Well, thank you for joining us on this deep dive into the fascinating world of gluconeogenesis and blood glucose regulation. We hope you feel a little more well-informed, maybe even amazed.
And from the whole Last Minute Lecture team, thanks for learning with us.